Okay, good afternoon, ladies and gentlemen. Good afternoon. It gives me great pleasure to introduce you to our final plenary speaker, Professor Susanne Foitzik. I don't know if she remembers, but I first met Susanna back in 2003. We were both based at the Universität Regensburg. She was undertaking postdoctoral work, and I was working on a master's thesis under the supervision of Professor Jürgen Heinze. Since that time, Susanna was appointed professor at Ludwig Maximilian University in Munich. She now holds a chair at Johannes Gutenberg University in Mainz. As well as publishing over 130 scientific papers on subjects as diverse as host-parasite coevolution, behavioral genomics of division of labor, and the behavior of slave-making ants. She has also co-authored, together with Olaf Fritsche, a popular science book entitled Empire of Ants, which I can highly recommend. Without further ado, please put your hands together for Susanne Foitzik, who will present her talk on coevolution and molecular regulation of ant behavior, chemical and life history traits. Thank you.
Thanks so much, Christopher, for the invitation. It has been a pleasure to be here. And I want to talk today about 3 different chapters of my work. So the first one is division of labor, as Christopher already said. Second one will look at the coevolution between social parasites, and here in particular, slave-making ants. Finally, I want to tell you a story of parasite manipulation. And most of my work I have been doing on Temnothorax ants. And these ants are tiny. They are very common also here in England. They live in acorns or sometimes under stones. And they have very small societies, only like 50 workers on average. And also the workers are small, only about 2 to 3 millimeters. But they're great models, and they are easy to collect and easy to maintain. And they have some features, as you will see, that allows to do manipulation, experiments with sufficient replication, and also there are now models for genomics. Well, let's start with my first chapter, which is on division of labor. Well, division of labor is an important organizational principle which occurs in all different types of biological contexts, as well in our human societies. It's also part of the 3 major transitions in evolution because it allows specialization of certain tasks, and it reduces the cost of task switching. And division of labor has been long known to be one of the reasons for the ecological success of social insects and also ants. Well, there is a primary division of labor between queens and workers, which obviously have different roles in the society. However, I have been concentrating more on the behavior specialization of workers, so that we have foragers and nurses, for example, in the colony. And there are many theoretical papers, as well as a lot of empirical studies in that. And one of the common model used is that of task thresholds. So the idea is that all ants or bees perceive an odor or a signal for a task similarly, but that they have kind of in their brain likely a different threshold whether they undertake the task. So let's say we have a larvae crying out for food. I mean, probably not acoustically, but chemically. And then some workers have a very low threshold, so they will smell this signal and they will, I don't know, do care for the larvae. Others have a very high threshold, and they will noticing the signal, but they won't do the work. And these thresholds were supposed to be influenced by different gene expression or genetic gene activity, which in turn is influenced by the age of a worker, by fertility, by nutrition, but also by colony demands. And when basically the stimulus is higher than the threshold, an ant would undertake a task. And because individuals vary, you get division of labor. So we started to study this with a first PhD student, Philip Kohlmeier, who now has a professorship in the US. And what Philip did is he first just wanted to see, do we have the typical age-dependent division of labor? As those people who work on social insights all know, normally young individuals stay inside of the nest and do brood care, and older individuals go out and do the risky task. The reason is you don't send out a young worker to do a risky task, because if she dies, you lose much more work for the colony, but you do it for the old workers, which anyway don't have a high life expectancy. And indeed, this is what Philip found. He found that young workers here in green do more brood care, but less foraging. And then he looked at a second behavior, which he called nestmate care. And this was grooming, feeding, and antenating adult workers and the queen. And here he found it was a bit more balanced. So both young and old workers were doing this. So it looked a bit like brood care, nestmate care, foraging. Okay. Well, then we wanted to know which genes underlie this behavior. The problem was we had age and task being entangled. So he wanted to disentangle this, and he did this by either removing all the foragers or removing all the nurses. And if you do that, then either foragers come back in if there are no nurses more there, or young nurses will go outside and become foragers. This way you can generate young and old nurses and young and old foragers. And this way he could disentangle the both things which are normally in a colony context entangled. And if he did that, he then analyzed the transcriptome. And we were actually surprised to see that more than 10 times as many genes were linked to the task, like whether an ant was foraging or nursing, than linked to its age or fertility, which he controlled for. And the most differently expressed gene he found was called vitellogenin-like A. Actually, there are multiple vitellogenins in the ants, and we found different copies of this gene to be upregulated in queens, in foragers, and nurses. Actually, when we look at the phylogeny of the different vitellogenins, you see in the upper pink section here, there is vitellogenin-like A, which we find upregulated in the nurses. It's actually a different copy than the vitellogenin, which is known to control honeybee nursing behavior. So in both cases, it's vitellogenins, but it's actually not the same copy. So we wanted to know whether this gene is really causative of controlling brood care behavior. So we did a knockdown of it by actually feeding a double-stranded RNA. And we managed to knock it down by about 70% in the fat body. And this was also interesting. I mean, we look at behavior here, but the gene is not expressed in the brain, but it's expressed in the fat body. So we knocked that down, and lo and behold, we could actually see a strong reduction in brood care behavior. So the knockdown is here in purple, and you see it's strongly less brood care behavior when we knock the gene down. So indeed, this knocking down this one gene controls a lot the behavior we see. Then we question, well, okay, they don't do brood care. Maybe they then go out and do foraging. We didn't see that. However, when we do a long-term knockdown, we saw that they increased their nestmate care behavior. So we get a decrease in brood care behavior, but an increase in the care for the adult workers and the queen. So when you think about it, they actually do the same behavior: antenation, grooming, feeding, but instead of targeting it towards the brood, they target it towards the adult members of the colony. So we were wondering whether they kind of are responsive to different signals. So instead of being responsive to the brood signal, they become responsive to the adult worker signal. Okay, so what did we do next? We extracted the cuticular hydrocarbons of either workers or of larvae of the same colony. We put them on filter paper, put the ant in the center, and said to the ant, well, tell us what you are interested in. And then we compared knockdown ants to non-knockdown ants. And we found what we expected. When we knock the gene down, the workers show less interest in brood odors and more interest in nestmate odors. So that fits to our interpretation of the data that indeed this gene controls the responsiveness of the ants to the odor of the brood versus to the adult workers. Okay, the question then we had was, what regulates the expression of this gene? And here we looked at histone acetyltransferases, because in Camponotus ants, there was a paper which showed that that might play a role. So we experimentally inhibited the histone acetyltransferase by feeding the ants with C646. And then we did the same experiment as Philip did at the beginning. So we either removed foragers or we removed nurses, and then we forced workers to switch the behavior. Well, feeding 646 didn't do anything if you didn't force ants to switch the behavior. If you took away half of the nurses and half of the foragers, we didn't see any change. So it wasn't directly poisonous. But if we forced them to switch, we got interesting switches. And what we could see is that actually when we take foragers away and we ask nurses to become foragers and we feed them with this inhibition of the histone acetyltransferase, they actually transition much faster than normal control workers did. When we did the reverse, tried to get foragers go back in to become nurses by removing the nurses, it actually impeded the process. So in both directions, we had a significant change, but it was not in the same kind of, in one way it was impeding, in the other way it was speeding it up. Our interpretation of the data is that the histone acetyltransferase activity keeps workers in the brood-carrying mode. So likely some important histones are acetylated of genes which are important to keep the workers inside and doing the nursing behavior. And maybe this is also important that young workers are not too early going out because the life outside is very risky. And we are now investigating which genes are directly targeting by the histone acetylation and have developed in our lab a cut and tag method to look at this. In the next study, we wanted to look more tissue specific. So we compared gene expression in the brain and in the antenna. And the antenna we did because we our work showed that it is a responsiveness to chemical cues. And obviously the antenna are the organs to perceive these cues. And we also looked at microRNAs. They might also have a regulatory function. Shortly to the microRNAs, indeed, we found strong differences between foragers and nurses in microRNA expression. We found several differentially expressed microRNAs, and we at the moment trying to investigate which microRNAs are targeting which genes and how are they involved in the regulation of gene expression here. But coming back to the RNA-seq analysis, where we compared gene expression in the brain and in the antenna. And when we did this, we were surprised to see that we find about, 8 times as many genes differently expressed in the antenna than in the brain. So I kind of expected the brain as a control for the behavior to be the most differentially expressed. No, it was actually the antenna. So we thought maybe worker behavior depends more on signal perception than at brain activity. And when we look closer which genes are differentially expressed, we found literally half of all odorant receptors to be differently expressed. So in this species, we have 419 odorant receptors. Ants had a radiation of odorant receptors because chemical communication is so important for them. And half of them were differentially expressed. And we found that nurses overexpress the co-receptor ORCO as well as ORs involved in the 9-exon gene family, which is known to be important for the perception of cuticular hydrocarbons. We found that foragers overexpress many different OR families, and that also makes in some ways sense because foragers go out and they explore, let's say, a diverse chemical environment and have to, found their way back, they have to find food sources and so on. You will see in a minute that these ORs are actually those which are convergently lost in dolotic slave-making ants, which don't do any brood care and foraging anymore because they outsource this to the host. And that also makes sense that these chemical perception are involved in regulating division of labor. So our data seem to show that the peripheral nervous system might act as a sensory filter, which might play an important role in regulating behavior. Sensory filters limit the information perceived by the brain. So instead of all workers getting the same information, some tune into a different radio, if you wish. And maybe the sensory filter might make a response threshold model superfluous. We are investigating in this at the moment. And we're also trying to investigate how is the filter set. So if we knock down VG like A, do we get different expression of odorant receptors in the antenna? And these are indeed ongoing experiments. I cannot tell you the results of that. When we look at odorant perception in these ants, we notice something else. We have an ongoing project with a neurobiologist, Carlotta Martelli, and a genomicist, Hugo Dara, and our students, Ina and Clarita. And this is investigating how odorant receptor information is perceived by the brain. And in the ants, in the brain, we have the antennal lobes, and in the antennal lobes, there are glomeruli. And in Drosophila, it's very well known that these 67 or whatever odorant receptors each have one glomeruli they compute to. So basically, we have one receptor type, one neuron type, and one glomerulus. And we kind of expected the same for our ants. However, when we investigated this, we found, and here you see a brain of Temnothorax longispinosus, and the antennal lobes are the little things below the two. And what we found when we counted the glomeruli is a strong variation in the number of glomeruli. So some individuals, some workers had 250, and others had the 419 we would expect. We are not quite sure whether this is now associated with division of labor, because we think actually that the brain is not as flexible as changing this number, but we're trying to investigate what the function of this is, and this could also obviously lead to different perceptive environments in these ants. At the moment, we also look how these ORs are expressed, and it's actually kind of an interesting thing to think about that each cell in the antenna have the same genome, but some of them have to express OR 420, and the other one has to express 280. And the question is, how is the communication done that you get all of them expressed in maybe equal numbers, but that not all, and when we look at this, we actually found that when we look at the genome, that the ORs are here organized in tandem repeats. And we are investigating how the transcription is done and how kind of the organization of the odorant receptors over the entire antenna is organized. But this is ongoing work. To sum up this chapter, I think we find a typical age polyism: young workers inside, old workers go outside. We see that gene expression is more linked to task than to age. We see that vitellogen like A controls brood care behavior and the responsiveness to social cues. When we knock this gene down, we see a behavioral progression from brood care to nestmate care, but not all the way to foraging. We can see that histone acetylation is involved in regulating the task switches, and we investigate in how. But we also see that our data show that the transcription activity in the antenna is important, which suggests that there might be a sensory filter where ants can tune in to kind of different signals and then concentrate on different tasks. And this variation in glomeruli number, which we found in the brain, we are investigating and see how and whether it is linked to division of labor or maybe has yet another function. Okay, first chapter. Now we go to the molecular coevolution of zoonotic ants and their hosts. Well, zoonosis, or slave-making behavior, evolved at least 10 times independently in the ants. And in my Temnothorax ants and their related genus Leptothorax, at least 6 times. So we have it again and again that they become these slave-making ants. So what is a slave-making ant? Well, here in red are slave-making ants. And since my ants live in acorns, I've drawn them nicely in acorn. So we have the mating flight, the queen flies out. She has to invade a host colony. She is obligatory dependent on the host. She has to drive away the workers, take over the nest. And then out of the worker brood of the host, the first workers emerging. They learn that this is their mom, and they do the normal works they would do in the normal nest. In the next year, we have slave-making workers emerging. but they don't do work. Actually, Darwin already cited this on The Origin of Species, where he said, it's fascinating. I mean, you can even give them food. If they're alone, they starve, they don't eat. They need somebody who gives them the food. They're kind of so utterly helpless without their slaves. And he was fascinating how you can get a loss of normal behavior in an ant. And in the next year, they will do raids, attack other colonies, steal the worker brood, bring them back, and basically outsource all the work, brood care, nestmate care, and foraging to the workers of this related species. Okay, obviously this puts a strong selection on hosts to develop defenses against this exploitation. It's not in the interest of them. And then the evolution of counter-adaptation. And we have been investigating this escalating coevolutionary arms race several years. I want to show you shortly here. This is from one experiment we did a few years back. You see a raiding column here of T. americanus slave makers going to the host nest of Temnothorax longispinosus, which was the species I did the division of labor. And you see how they come, and what they want is not killing the host. They want only the worker pupa, because when they bring them back, they can enslave those. And I mean, you will see there are more coming. There's really quite a raiding party, and you will see there's a lot of fights going on here. The hosts don't give the brood lightly. I mean, they know that this is their enemy. We did studies and show that they really recognize their enemy. So, no, I can. So a few minutes later, you see nearly all the brood is done. The host tries to rescue the brood. The slave maker tries to take it home. So we looked into how did the losses evolve, which traits co-evolve in social parasites and the hosts. We recently did a study where we compared 3 slave-making ants and their hosts, and we found well, you know, it's general that parasites often lose genes. So here we have a social parasite. Do they also lose many genes? Have they a reduced genome? Was the question. Well, they don't need anything which is necessary to do brood care behavior because they don't do that anymore. We also found that they have a different chemical profile. They have fewer methylated cuticular hydrocarbons, which are important for recognition in the host. So basically, they are a bit mystery ants. They don't smell a lot for the host, and this might help them to invade the nests. But when we look at the genomes of these 3 independent origins of slavery and the host, we found that they lost 50% of their gustatory receptors. So, okay, while slave makers might get the food brought by their slaves, they might not be able to taste a lot when we look at the loss of the gustatory receptors. When we look at the odorant receptors, we found that 30% of those were lost. And we found that the same odorant receptors were lost in each of these 3 independent origins of slavery. And these were actually those odorant receptors I showed you before to be differentially expressed between foragers and nurses. This makes sense. And actually, in a more recent paper where we now have a 4th origin of slave-making behavior, we confirmed this loss of odorant receptors, and we also could show that they lose those odorant receptors which arose very early during the evolution of ant sociality, which is basically those which we know are differentially expressed between foragers and nurses. This comparative study of the 4 slave maker genomes and 4 host genomes within the genus Temnothorax showed that actually social parasites and hosts have similar genome sizes. So we don't get a strong reduction in genome size. However, we found that social parasites have more protein families under both positive and relaxed selection. So relaxed selection makes sense. Everything you don't need anymore because you're a social parasite might be under relaxed selection, but there are protein families under positive selection. We also found that parasites retained more de novo genes, so genes emerging from non-coding regions. Problem is a bit, we don't know what these genes do because they are de novo. We can't copy down an annotation, obviously, because they're new. So we are investigating further what that means. In another study, we wanted to see how slave makers influence the behavior of the slaves. And what we did here, and this was here on the European system of Haparoxenus sublevis and its host Leptothorax azaborum. So we took worker pupa, and we either put them back to the host nest, we put them to another nest of the same host species, or into a slave-making nest. We let them emerge and do their work for 6 weeks. And then we sampled them and look at gene expression, both in the brain as well as in the antenna. And we found, especially when we look at gene expression in the brain, there is a different gene expression. In yellow, here are the slave-making nests. There is a different gene expression in workers which are enslaved, if you wish, compared to non-enslaved workers, even when we compare it to non-host, host nest of a different host nest. We also had this when we look for some gene, when we look at the antenna, but also there we found that actually there is an effect of whether you are working in your home nest or in a foreign nest, independent of whether that was slave-making ends or not. And again, we found that odorant receptors are important and that the worker behavior in the hour before we sacrifice them was strongly linked to gene expression in the antenna. So again, this odorant reception seemed to be important to regulate behavior. But now come back to my mainly investigated system, that is the slave maker Temnothorax americanus and the host Temnothorax longispinosus. And they occur in the United States and Canada, from really Montreal down to West Virginia, east to Illinois. And we sampled many different populations, and we wanted to see whether we find traits in the hosts which are linked to parasite pressure. And we did find that. We found that host populations from Temnothorax longispinosus are more aggressive if there are slave makers around. They actually flee rather than fight when they encounter a slave maker. And this is because in highly parasitized sites, slave-making colonies are large and they have large raiding parties. So the hosts don't put up a fight. When they detect them, they just grab the larvae and they run. Whereas in less parasitized sites, the slave makers have smaller colonies, and there they try to fight them, sometimes with some losses. We also show that division of labor is associated with them. They exhibit more diverse chemical profiles when slave makers are there. Maybe the host tried to have different profiles so that the slave maker has a problem to match it. They are less responsive to the Dufour gland manipulation. And finally, we could show that sometimes enslaved workers kill the slave maker brood, and we showed that this could be interpreted as a case of slave rebellion. But we also found that the slave maker develops counter defenses. We have field manipulations show evidence for local adaptation, and we found that slave maker populations which carry less recognition cues on the cuticle are more successful in invading the colonies, and in that population we have more slave makers relative to hosts. With this, we were interested in a new study to investigate the genetic basis. And now we don't use microsatellites as we did before, but we resequence the genomes. And we wanted to see SNPs which are linked to the coevolution between slave makers and hosts. So we did a large-scale study by collecting in 10 different sites, and we collected always 100+ host colonies and as many slave-making colonies as we would find, which was 8 to 80, so it varied. We maintained them in our lab for 1 year under standardized conditions. And we wanted to see first, how is their behavior and how is their chemical profiles, but then also trying to get genetic variants linked to behavioral chemical differences, as well as to parasite prevalence and also to climate. And this is also something new we did. We looked at the climatic conditions in each of these 10 sites. And we found that they actually linked. So in warmer sites in the south, we found more slave makers. In northern sites, we found less slave makers. In our statistical analysis, we always try to disentangle as much as possible parasite prevalence and climate. But in some case, it was still entangled. I just have to say that. Okay. No, this I already said. I have this twice, sorry. I'm clicking through this. Yes, okay. The first study we did looking at the genetic basis of host-parasite coevolution was this one. And we used a pool-seq approach in this study. So what we did is we took 100 workers from 100 nests from each of the 10 host sites, and we did a pool, and then we sequenced at high sequencing level. And then we did a GWAS analysis, a genome-wide association analysis, to identify single nucleotide polymorphisms associated either with parasite prevalence or with climate. And we did, and we found 288 genes with SNPs linked to parasite prevalence. And here you see the enrichment of those genes which are linked to parasite prevalence. And you will see peptidoglucan catabolic process as the most enriched term. This was surprising because the peptidoglucan process is involved in antibacterial defense. It actually recognizes the bacterial cell wall, and it was like, okay, but this is a social parasite. Then we found innate immune response. Again, okay, immune, I mean, these are social parasites. And finally, sensory perception of smell. Okay, that I get. But you will see that this peptidoglucan catabolic process will pop up again and again. So there seems to be a pattern, and we're trying to get what it means. We found non-synonymous SNPs in odorant and gustatory receptors, and when we looked at the SNPs linked to climate, also in heat shock proteins. We also looked at the traits expressed in our 100 workers pool, so a kind of population-specific traits. So we looked at the cuticular hydrocarbons of these 10 populations using the same workers where we did the genome sequencing for. And we found that actually the cuticular hydrocarbons profile was linked to climate, but not to parasite prevalence. We then tried to look at the genetic basis of these cuticular hydrocarbon variation. And here it was interesting. So we look at signaling, CHC production. And when we look at it, we find SNPs in odorant receptors to be linked. So in signal perception rather than signaling. But also in fatty acid synthesis genes, which makes more sense. And finally, in this study, we looked at the expression, the gene expression in the antenna again. So we put 100 antenna from 100 workers, from 100 colonies, from each of the 10 sites. And we found that gene expression in the antenna was linked to parasite prevalence, and quite strongly so. So obviously, what is happening in the antenna is probably under the selection by these slave makers. And we are investigating this first. The next approach we took, we now did resequencing of individual workers. And here we took 15 workers from 15 colonies per each of the 10 sites. And we did this for the slave maker and the host. So now, and before we did behavioral analysis and chemical analysis for these 15 colonies per population. So now it's more of an individual or colony level based approach, not population based approach. We again found SNPs related to parasite prevalence. And here, this is a picture for the Manhattan plot for the host. And using this approach, we found 20 odorant receptor genes with non-synonymous SNPs. But again, we found 17 peptidoglucan recognition proteins. So it's a different approach. I mean, same populations, but a totally different approach. And we found it again. You see everything which is red are these PGRP genes. So something is going on there. Together with the Finnish colleague, we are investigating this further because she could show that these genes radiated in temnothorax compared to other ant species. So they have about 30 different genes, whereas in other ants you have only 8 to 10. And in the slave makers, there is a reduction again. We don't know whether they have now a different function, but this is the pattern we see. We also found SNPs in fatty acid synthesis. So basically what we see, odorant perception, antibacterial defenses, okay, I don't know, and cuticular hydrocarbon synthesis. So signaling, perception, and immunity. We looked at the slave makers. We found 2 odorant receptor genes in the selection under the slave makers. And we did the same also for the climate. I didn't show you all the data. And in the climate, we actually found SNPs in the same genes in host and parasite. Whereas when we look at host-parasite coevolution, we didn't find SNPs in the same gene. So they seem to use different strategies of offense and defense. And then we tried to link the genomes of these ants to their traits. So what we did is conducted 15 times 10, so 150 behavioral experiments times 3, where we either introduced a nestmate worker or a non-nestmate worker or a slave maker from the same population into a colony. And we videotaped and analyzed the behavioral responses. And what we know that the behavioral response is important in the coevolution between them. And we wanted to get to the genetic basis of this. Well, first we found that hosts aggress slave makers more than host workers. So they know their enemy. We also found that hosts are more aggressive in colder, more humid sites. but as well as in sites with higher parasite prevalence. Hosts pick up the brood and flee more often when they see slave makers, and especially so in warmer climates, so which is in the south where slave makers are also common. Interestingly, we found the opposite for the slave makers. So in the north, the hosts were very aggressive when it was cold. But in the north, the slave makers are less aggressive. So it's actually the opposite, which could maybe explain why we have more slave makers in the south, because here the hosts are not aggressive, but the slave makers are. Yes, and they encounter in colder regions, slave makers show more passive and submissive behavior and apparently are not so successful. So trying to get the genetic basis of these behaviors, we looked at SNPs associated with host aggression. And here we found the response to axon injury, toll-like receptor, and visual perception. But we also looked at SNPs which are linked to the responses of host workers to injured nestmates. During raids, host workers sometimes lose limbs like legs or antennae. So we simulated this and we put an injured nestmate back, looked at the behavior, saw that the behavior was linked both to parasite prevalence and to climate. And then, because it was done on the same colonies where we resequenced the genomes, linked this to, and here we found odorant receptors to be important, as well as fatty acid biosynthesis. Note again, signal perception and signaling being linked to it. Parasite submissive behavior, we found also an odorant receptor to be involved. But going back to this peptidoglucan recognition thing, which, so here that was a top GO-enriched term when we looked at host adaptation to parasite prevalence. And we found that the allele frequency in 104 variable host loci within 11 different peptidoglucan recognition proteins were linked to parasite prevalence. So something is going on with these genes. I just don't understand what they really do. And this is something we have to investigate further because we did not expect this link. We looked at the cuticular hydrocarbons, the signaling as well. We found that the CHCs in the host was linked to climatic conditions. In the parasite, however, it was linked to parasite prevalence. So for the parasite, apparently being able to invade a host nest is more important than the climate was. We did, again, our GWAS analysis. And again, we found this, that the cuticular hydrocarbons trait was linked to odorant receptors as well as fatty acid biosynthetic process. So again, when we look at the signal, the CHCs, we found odorant perception, SNPs in odorant perception genes to be linked to signaling, as well as to the genes which generate the CHCs. So to sum up what we did on the coevolution between these douletic ants and their host, we found that slave-making ants convergently lost odorant receptors and also gustatory receptors, which are critical for worker tasks. We found that social parasites can influence the transcriptional activity in brain and antenna of enslaved workers. Host and behavioral and chemical defenses are linked to parasite pressure, and the internal transcriptional activity seemed to be linked to parasite prevalence. So again, it looks like the signal perception in the antenna plays a role, not only in division of labor, but also in host defense against parasites. But we also found an astonishing strong effect of climate, which is linked to chemical and behavioral traits. So before we always looked at parasite pressure, now we know that climate is not unimportant here and co-varies with parasite prevalence. Parasite prevalence in host and parasite are linked to SNPs in genes which are important for odorant receptor genes, fatty acid synthesis genes, and peptidoglucan recognition genes, so linked to smell, signaling, and immunity. So the genetic basis on co-adaptation involves these three aspects, and we're investigating, obviously, this currently further. Okay, the last story I have for you is a story of parasite manipulation. You know that many parasites manipulate host behavior, and we love this kind of stories of like the Lancet liver fluke who changes ant behavior to bite into grass stems. Why do they do this? Often to improve the transmission to the final host. So these are often parasites with complex life cycles. They use not one host, but two or three even. And we are investigating how is this possible. And our model system here is the cestode Anomotenea brevis, and the ant Temnothorax nilanderi, and the woodpecker. And there are different woodpeckers who are important. And the life cycle goes as this. The tapeworm is in the woodpecker, which defecates, and with the cestode eggs are released, and for whatever reason, they are attractive to the ants, and they take them home and feed them to the larvae, and the larvae get infected then. Once the infected workers emerge, they look different. They have this kind of yellowish cuticle, and it's much softer, so the cuticle is less melanized and less sclerotized. And then, well, then eventually the woodpecker has to come along and to eat the infected ant. And the cestode lives actually not in the gut, but lives as cystocercoid larvae in the hemolymph of the ant, loosely attached outside to the gut. And an ant can have between 1 and 73 cystocercoids. So it's not, so that varies. Okay. So the first thing which we could show is that infected workers are smaller and they show different behavior. They stay close to the brood. They rest more and they work less, but they are more often fed. So the others care for these infected workers much more. They remain in the nest even when they are disturbed. When I open an acorn, everybody runs away except the infected workers, which are like, huh, okay. I mean, obviously, this could increase transmission to the woodpecker when it opens the acorn. They also show an altered, less diverse microbiome. We also found that workers of that uninfected workers living in infected nests show an altered behavior. They are less aggressive. And if I take infected workers away, they become more aggressive to non-nest mates. And if I put them back, the aggression goes down. It's really fascinating how you can manipulate that. While Sarah, my PhD student, was doing this study, she came to me like, something is wrong with these infected workers. They never die. I was like, what do you mean by never die? And she was like, look at my data. You know, the yellow ones are the infected ones. And over the 4 months, I basically had no death of them, whereas the others died at higher rates, and especially those which live with the infected ones. And I was like, okay, well then maybe investigate this long term. And she did. She looked at that for now 3 years doing her PhD. And these data confirmed her original observation. So what she found is that infected workers actually show the same survival rate as the queen. There was no difference. And queens in the species can get 20 years. And when we started the experiment, the infected workers were already adults as well as the queens. And then we marked newly infected, newly emerging non-infected workers, and we observed their survival. And we found, well, after 3 years, all of them were dead. And we find again the difference we find before. If you live with infected workers, you die at higher rate. Okay, so having a cestode is actually nice for your lifespan. You live longer. You live actually 4 to 5 times longer. And a colleague actually told me, oh, by the way, I had one of these infected workers for 7 years. And I only stopped doing it because I didn't want to care for the colony anymore. She didn't die. I was like, okay, so they can live really long. Actually, we reanalyzed this data recently, or better, Victor and Hannah Coco did that. And she could show that when a queen, which is not infected, lives in a nest with infected workers, the more infected workers are there, the shorter the queen lives. So also the queen pays a price. So yeah, when I'm infected, I live longer, but my colony is not necessarily happy. The queen actually has a higher death rate, especially if it's many of us. And we also found that for infected workers, if there are many, if you have up to 70% of infected workers, which we sometimes have, they die also at a bit higher rates. So it's best as it for an infected worker if she is the only one in the nest. We also found that the sex ratio changes. So infected colonies produce more intercars and also produce more males. So somehow the colonies pay the price. The individual, however, has an expected an increased lifespan. Okay, a tapeworm is acting here as a fountain at views. Okay, let's try to investigate how is it possible that they live so much longer. So, I mean, I already told you it's still a parasite on a colony level, but on the individual level, we see this increased lifespan. Okay, so the first question we were like, okay, maybe infected workers are kind of physiologically like queens, you know, because queens can live 20 years in the species. So we looked at the metabolic rate, and actually here infected workers match young workers, but not the queens. They have a higher metabolic rate, and they also have a higher lipid content. However, they get more social care. They get more social care as sometimes even the queen. So basically, somehow they are attracted to the other workers. So what we did is we looked at the cuticular hydrocarbons, we extracted them, and we presented either infected worker cuticular hydrocarbons or non-infected worker cuticular hydrocarbons to workers in infected nests. And sure and behold, they loved the cuticular hydrocarbons of infected workers. So somehow they signal care for me and they get the care. And the queen pays a price for that reduced care. And they do have a different chemical odor. So what we show is actually infected workers seem to remain in a somehow young state. So they are not queen-like, really. There's some overlap with the queen, but not generally. We found, for example, that they are more resistant to oxidative stress. Here we used paraquat to exceed oxidative stress, and we find a normal reduction in survival with normal workers, infected workers. We didn't find an increase in mortality. So somehow they can deal with oxidative stress. When we remove the queen, infected workers develop their ovary and are the first to lay eggs. So certainly the lifespan expansion is not by castration, but rather they can develop their ovaries. They are fine with this. So who benefits from this manipulation? We think benefiting is actually the testote, because he, or it, I guess, wants to reach the final host, the woodpecker. And if I can extend the lifespan of infected workers, I don't know, by several fold, the likelihood that eventually the woodpecker comes by and eats me will be increased. So the parasite could be here a puppet player. And we wanted to look on a molecular level, how is this possible? The first study we did is looking at brain gene expression, also because we have these behavioral changes. And here we find indeed that infected workers upregulate genes which functions in the fight against oxidative stress. We also found that they downregulate muscle genes. And here you see a picture of the muscle, the mandibular closure muscle, and you see gaps in the infected workers and no gaps in the uninfected workers. We don't know, because this wasn't age controlled, whether this is because the infected workers were so old, but it can explain why they're so inactive. I mean, if you have no functional muscles, I mean, it makes sense. Then we compared gene expression in the guster between infected workers and the queen. We didn't find more overlaps than expected by chance, but one gene was interesting, and this is carboxypeptidase B-like, and this is in Drosophila known as the silver gene, and this is linked to postponed senescence, and was upregulated both in infected workers and the queens. Whether this now really is part of the story, we don't know, and we are investigating. The next study was now trying to really see what the testote is doing. So what we did is we took the hemolymph of infected workers, of non-infected workers, and infected workers from non-infected nests, and we also looked at the proteome of the testote. Now we look at the protein level. So here you see the comparison between infected workers and their nestmates, and everything in red is actually testote protein. So we identified 234 testote proteins in the hemolymph of the ant, 98 in every replicate. And actually 7% of all proteins that we identified in the hemolymph were from the testote. So if imagine that in your body, 7% of your proteins in the blood are actually from a parasite, I think this is a rather high fraction. We also found that the proteins from the ant are different if it's infected or not. And here we did see similarities between the proteins found in the queens and proteins found in the infected workers. But coming back to the testote proteins, 34% of those proteins had a secretory signal. So it looks like they are released maybe by vesicles, extracellular vesicles, because we found evidence for this as well. Okay, what kind of proteins are these? And now, yeah, we are a bit handicapped by this not being a model system. We have the genome of the testote by now, but still, because the next genome is so far away, a lot of the proteins we couldn't annotate. I mean, we could annotate actually 85% of the proteins we identified in the cystocercoid itself, but we looked at the proteins from the testode in the hemolymph, it was only a third. And especially those proteins which are released are probably novel proteins, maybe shaped to really manipulate this host. We didn't find an annotation. So you see here the 50 most released proteins. Everything in green we have an annotation for. We started now to use RNAi to downregulate the expression of the first 231 protein, that is the highest one, in the end. So in the testode in the end, and we managed to do that actually via RNAi, and then we did transcriptomal studies of the fat body of the end, and we found that a handful of genes changed their expression, among them 3 cytochrome P450. And we are currently investigated what could be the role of those. The proteins which we could annotate were some involved in gene regulation, others in oxidative stress resistance, such as thioredoxine peroxidase, as well as superoxidase dismutase. So the testode is apparently releasing antioxidants into the bloodstream of its host, and this in part could maybe explain the extended lifespan. But we were also interested in how is parasite load affecting gene expression. And here's a study of Tom. So what he had uninfected workers with 1 cestode, and workers between 18 and 57 cestodes. And when we did this study, we found that some genes are upregulated in infected workers or downregulated in infected workers, but others we found a kind of a doses effect. If you have more cestodes, they go more up in the host. And this were especially genes which have a response to oxidative stress. So not only does the testode releasing antioxidants into the end itself is upregulating antioxidants in response of having many infected testodes. We also looked at the testode gene expression, and here we found that if testodes live together with many other testodes, they upregulate genes which are involved in immune avoidance, as well as genes which are important for the fight against starvation. So if you are with 73 other testodes in the hemolymph, you have a problem getting not enough nutrients, and you have to avoid that the host is recognizing you and encapsulating you or whatever. When you are, however, the only testode, we found an increased expression of vesicle-mediated transport genes. But then this is a social host. So it's not only how many testodes one worker has, but it's also how many testodes, how many infected workers you have in the nest. As far as we know, they get infected during the larval stage, and all infected workers with the yellow cuticle are indeed infected, but they can't transmit it between themselves. But we have sometimes colonies with up to 70% infected workers, and other cases only 1. And we wanted to see whether that affects gene expression. And it does. So we see actually a strong change in gene expression when you are the only infected workers or when you are with many others. We see also an effect on the testode, even on the uninfected workers, which, when there are many infected workers in the nest, show a decreased survival rate. So here it looks like the molecular physiology of ants and cestodes are influenced by the prevalence of infected workers, as is green survival, which I showed you at the beginning. Well, if testode proteins target end gene expression, then there should be links. And what Tom did is now, he had 15 samples where he had the gene expression of the testodes of that worker, as well as the gene expression of the fat body of the host. And now he put all the data into one WGCNA to look at statistical links. So if we have a gene, for example, the superoxidase dismutase, where we know that this protein is released into the end. So if a testode upregulates this, do we see corresponding changes in gene expression in the host? And he could find these links and found that these genes are involved in cellular metabolism of the host. So we're trying to really get kind of like a detective. What is the testode doing? How does it change things in the host? And how does it change the life expectancy, what we see? We also looked at whether infected workers have a different immune response when we challenge them with LPS, which simulates a bacterial challenge. And there it was interesting that most genes change the expression with whether you are infected or not. We also had a response to our LPS immune challenge. but there was no interaction. So that means infected workers are as equally able to upregulate the immune genes when they're challenged by a bacteria. Interestingly, however, we found that there was a strong overlap in genes. So we found 114 genes, and for every single one of those, they were downregulated in cestoid-infected ants, but they were upregulated when we give a bacterial challenge. So these infected ants kind of have still the ability to get an induced immune response, but it is in part the same genes which are involved in this. So to sum up this story of an ant, a cestode, and a woodpecker, which I didn't talk so much about, we are at an entomological conference. So the infection with the cestode induces changes in the behavior, in the cuticular hydrocarbon profile, in the morphology, and the lifespan of ant workers. Infected workers live longer. They show survival rates which are similar to that of the queen. Why? Well, likely the transmission to the definite host is increased if infected workers live longer. Infected workers remain somehow in a physiological young state when we look at body fat and metabolic rate. They maintain their reproductive potential, so we have no host castration. They receive more care from their nestmates, and they upregulate longevity genes such as transferrin, carboxypate B-like, also antioxidants. And they're affected by parasite load, and this both on the individual level as well as a colony level, because we have a social host here. And finally, the cestode is releasing proteins into the host with gene regulatory functions, some are antioxidants, and with a lot of unknown functions where we are currently exploring further to see how is really the parasite manipulating its host. And with this, I would like to thank a lot of co-workers to help me to do all this study. Obviously, I received some funding from the German Science Foundation, and I have currently one open PhD position. So if you are interested, especially in the division of labor project, please send me an email. You could start in January. Okay, thank you.
Thank you, Susanna, for a fantastic talk. Really interesting stuff and a huge amount of work too. So do we have any questions in the audience? Yes, the gentleman up there.
Thank you so much for that. I was wondering with the slave-making ants, are there any systems where adult workers are taken as slaves, or is it absolutely necessary that they're taken before they're adults to tolerate or accept being slaves?
Yes, there are some. So one of the species which we described only about 10 years ago, Temnothorax pillagens, there are two types of raids. So they try to actually go into the nest and undergo the radar of the host. And if they manage to do it, they encourage the host to either relocate to them or they relocate in the nest. And actually my PhD student, Amir, just told me, this one raid was really weird. They just moved in with the host and the host didn't even notice. I was like, yes. But if the host does note it, we saw that this slave maker is actually quite radical. So it could be within 10 minutes, they sting every host to death, every adult host. So they have this kind of two sides to it. If they manage to stay under the weather, they do that because then they get many more workers. I mean, it's better. And we also have Temnothorax, or it was called Myrmoxenus ravui, Temnothorax ravui. And here the queen is invading the nest. And I actually observed that during my PhD. She tries to kind of stay at the side and not being, she's actually quite small. And within 3 weeks, she went to the neck of the host queen and throttles her. I mean, obviously, she cannot put the oxygen away because, I mean, they have trachea, we all know that. But maybe she does something with a nerve cord there. The host workers don't seem to notice. And after 3 weeks, the host queen was dead and she was replaced her. So that is sometimes possible, yes. Also, the polyagous, it occurs. In Americanus, I never saw that. Actually, they often fight. And when we collect those colonies, only with a slave-making queen and the host workers, they often have injuries. Like a third of them have lost antenna, the queens. And in one case we collected this year, she had 1/2 antenna left, and I think 1.5 left legs on the left side and 1 on the right side. But she was happily living in the colony. The host workers were doing the work. She was laying eggs, and they must have a very good healing mechanism because, I mean, obviously these wounds had to heal, and they did heal in this case. So yes, there is both strategies possible.
Any more questions? Yes, gentleman up there in the blue.
Thank you so much. Amazing talk. Reeling over it right now. A very simple question, probably a bit naive one. Have the ants come up with any form of like counter defense against the parasites, like the tapeworm parasites? Any behavioral ones, maybe?
So we did find, I mean, infected workers smell slightly differently. And we did find that they, as I said, they get more attention, but we also found that they are more often getting slight aggressive responses. So we see workers intonating them, and then they open the mandibles, and sometimes they even slightly bite. And you're like, they do notice something is different, but somehow they don't kick them out. And I think because they have some kind of pheromone which decreases the aggression of the non-infected workers, so they are maintained. We have not looked at the cestode eggs. So you would expect that maybe some of these host colonies start to not take, I don't know whether they really work the bird shit or whether they really are attracted to the cestode eggs. Obviously, it's in the cestode interest to make the eggs chemically attractive to the ants, but we haven't studied that. At the beginning, I thought, okay, the costs are not too high, but now with the new data Hannah analyzed, showing that the survival rate of the queen really goes down. I mean, on the colony level, this is a parasite, you know, and it has fitness costs.
We've got an online question here from Bikash, and they say, amazing talk. Are odorant receptors reduced or less prevalent in reproductive individuals such as queens and males due to their lack of involvement in foraging activities?
We haven't studied it yet. I know that there are less glomeruli in some ant males. We haven't studied it ourselves, but this has been shown. I don't know about queens. We are currently looking at this variation, and we also don't know whether the variation we see in longispinosus, that some have 250 glomeruli and others have up to 420, whether this is related to body size in any way. So we don't know that. I mean, the genomes obviously have all the same number. I mean, this is the same, but when we look at the glomeruli, there might be variation, yes.
Mm-hmm. Any more questions? Yeah.
Hi, sorry, that was really interesting. I might have missed this, but do sort of pathogens and other parasites have any importance in the sort of slave makers with all these colonies interacting with each other and stressing each other and the differences in temperature and those kind of things?
We haven't studied it yet. I mean, this peptidoglucan protein family obviously did a radiation in the host. We don't know what the function, whether it's still immune defense. The function is hosts are more likely to be injured in areas where slave makers are around. And we saw that they have a stronger response to injured nestmates. We didn't look at the immune response. For the slave makers, I really would anticipate that, because at least in T. americanus, I see all these formerly injured individuals, and I think they must have a strong immune system. So we should investigate this. And as I said, this is peptidoglucan gene family. It's an unexpected result, and we will go into that further. That's a really new, unpublished result.
And does Luke have a question?
No,
oh, okay.
Okay.
Any further questions? Yeah.
Given that the slave makers seem to be so completely hopeless at self-care, is it possible that they're just filthy? That they're just bringing all these like fungal spores and bacteria on their surface when they go raiding or when they bring the host back to their nest? And so the host is responding by cleaning them up, essentially.
I mean, the slave makers only go out during the raiding season in summer. It's about 6 weeks, the time when the hosts have the worker pupa. This is the time they do the raids, also when it's the warmest days. We have not investigated. We neither looked at the gut microbiome, not at the viruses or any of this sort. We haven't investigated, so I cannot say. It is. I mean, we see a lot of grooming, and the hosts are grooming the slave makers a lot. But we don't know whether this is because they are filthy. I can really not say. We haven't looked at viruses, bacteria, anything. Yeah, no, don't know. But interesting idea.
And
okay.
Thank you for the great presentation. I was wondering why the lifespan of Kuhn is shorter. Is it because she gets less care from the workers, or is the chemical environment changing, making it less feeding or things like that?
So we find a decreased survival of non-infected workers and also of the queen when there are many infected workers, yes. And I think indeed it is because they get less care. For the queen, it was clear that there was an association between, and we actually also saw when we look at the behavior, how often a queen is groomed and fed. It depends clearly on the ratio of infected to non-infected workers. And with more infected workers, they get less care and they survive as well. So care seems to be, likely the chemical profile of infected workers signals care for me, and they do that. I mean, it depends a bit on the composition. When there are many infected workers, the queen really gets very little. If there's only one, the queen seems to be still doing okay. But as it increases, it's really getting a problem. Mm-hmm.
And yeah.
Yes, thank you for such a fantastic talk. This is a really interesting effect with the pathogens impacting behavior. And you mentioned there were a lot of other slave maker ants. If you were to try and investigate this effect in another slave making species, what species would you go for and why, do you think?
Well, basically, there are two different main clades where slavery arose. And we focused here on the myrmecine ants and actually the formicine family, Temnosora, Cleptothorax. But there are also slave makers in Formica and the wood ants. And there are also facultatively slave makers who Formica sanguinea, can live with and without slaves. And would be interesting to see what happened then. When I expect that they wouldn't have such a strong signal of odorant receptor loss because they still are able to do the work, whereas here we don't find it. They don't do it anymore. So that it might be interesting to look at this parallel evolution yeah. And as I said, with the immunity, we really have to go further there. We haven't really had this on our radar. I mean, signal perception and signaling, this I really, because here the parasite is recognized by its chemical profile with the odorant receptors expressed in the antenna. So this we had on the radar. These immune genes were a bit surprising.
Brilliant. Thank you very much.
Okay, if there's no more questions, could you put your hands together for Susanne? So, on behalf of the conveners, Chris, Michelle, and Sharon, I'd like to thank you all for what's been a very interesting and productive conference. I said at the start that I hoped it would be an edifying and convivial conference, and I think it's achieved that. Just to let you know, you'll see up on the screen there that next year Ento25 will be in the University of Strathclyde in Glasgow between the 9th and the 11th of September. Don't forget to publish all your interesting work in society journals. And I'd like to thank the RES staff, in particular, Amy Everard, for all her help. So if we could give Amy and the rest of the RES staff and the student helpers as well, who've been going around. And now I'll hand you over to Professor Jane Stout, who's the forthcoming president for the RES. Thanks, to close the meeting.
Thank you very much. And the first thing I wanted to say was a big thank you to the RES team and particularly Amy. So you stole my thunder on that one. But the second thing to say is thank you very much to the local conveners for a fantastic scientific program. It really has been an amazing couple of days. It's been brilliant to hear so much incredible insect science going on. I want to particularly thank the plenary speakers with the just amazing talks. And I think, you know, that those of you who are earlier in your careers will look at these talks and see how much work and how long it takes to be able to tell these stories. But everything that we do and every little piece of work we do, whether as a master's student, a PhD student, all of that contributes to this body of knowledge that then tells these amazing stories. So thank you to our plenaries. I can't see the other two, but thank you, Susanne. Of course, the other thank you I want to say is to the RES staff. I've now officially taken over as the president for the society, and it's something that I wouldn't have imagined doing a few years ago. And it's fantastic to meet such a great team that run the RES so very well. It really is. It's a pleasure to work with you all, and I'm looking forward to the coming 2 years. The final thank you is to all of you for attending, presenting, enthusiastically asking questions and networking and talking to each other. And I hope you agree it really has been a fantastic couple of days. And I really hope to see you all next year in the University of Strathclyde in Glasgow. And as Chris just said, until then, keep doing the science, keep publishing in the RES journals, and I hope to, as I say, see you all again next year. So thank you very much. I've actually just remembered one really special thank you that I forgot to say, which is thank you to our former president, Jane Hill. Glaring admission, but thank you, Jane.